This is 2016 smoked ham. Take a lighter to the needle and sterilize it before I take a sample. But I'm going to put it on this one right here. I already wrote 2016 smoked ham, ice cream pig, ? On the bottom, because that is what it is. If you haven't figured it out yet, the reason I am using needles instead of Q-tips like I did in the last experiments to infect my cultures, the reason is because it is much easier to sterilize a Q-tip. You just use a flame like this, and a Q-tip becomes very sterile. I mean, a needle becomes very sterile. And that way, there is no bacteria besides the ones that you rub it into getting into the culture. So I slashed the surface of the bacterial culture multiple times with the needle that had been sterilized and then stabbed into the pig. I'm actually going to take a swab of is dog culture V2. Even though I didn't necessarily do the best job with it, I really want to see what bacteria I can grow from dog culture V2 onto one of these cultures. So yeah, I'm gonna sterilize another needle with my lighter, and I'm gonna get to work. Okay, so as you can see, I took dog culture V2, I took the thing out, I sterilized the needle, and now I have made dog culture V3. One thing that is very important to remember about this is the main thing that I did with dog culture V2, it melted. So the stab culture melted because it got too hot, so it didn't actually separate the motile and the non-motile bacteria species. But what it did do is, since it was exposed to an environment without any oxygen for such a long period of time, and since I took out the Q-tip and I swabbed the very bottom of the Q-tip with a needle, I did get rid of any aerobic bacteria, which are bacteria that can only survive in an oxygen environment. And I am only left with anaerobic bacteria and fossilitive anaerobes, which are bacteria that can live without oxygen, but they can also breathe like aerobes when in presence of oxygen and breed faster. So yeah. Finally, I went down to my cat's litter box, took a little sample, and stabbed it with a needle. So yeah, this is all three of my cultures. The dog culture V3, this 2016 smoked ham culture, and this litter box culture. So, filming this on November 9th. You might be wondering why you're not seeing it then. And the reason I'm not showing it to you guys until a couple weeks ahead is because of the fact that I've already got a lot of videos and I am going to have to wait a long time for the bacteria to grow since I am growing it in a refrigerator. That means that it is going to take 4 weeks for the bacteria to grow fully. And you might be wondering why I'm growing it in a refrigerator, and to learn that, you will have to watch the next video. So, hope you enjoyed, and see you then.